We are really excited to have three fantastic speakers this morning, or this afternoon. Clive Meanwell, Nevan Krogan, and John Sello. And because they have so much to say, I don't want to waste a lot of time introducing them. What the format of the morning is, each of the speakers are going to give a lightning talk, 5 minutes. And after that, we're going to have questions. Send in your questions, and I will then direct them for the last 15 minutes. And then magically, our session will be disappeared by the program organizers. So without further ado, I'm going to stop sharing. And Clive, I know you have so much to say, so take it away.
Thanks very much, Laura, and thanks to CERSI too. Useful clinical trials come in three models: difficult, very difficult, and extremely difficult. The first two estimate truth about what just happened in a medically and behaviorally controlled framework. The third estimates truth about what might happen when that control is removed. Difficult ones are near laboratory experiments. Sending doses of something or other is given to 40 courageous people to estimate how they handle the drug and how the drug handles them. Trained staff stand next to patients, surfing Instagram for the day, and they cost about $1 to $10 million. Very difficult ones look for drug effect. The good
Clive,
ones.
are you sharing your slides?
I don't have any slides.
Oh, okay.
Good ones randomize educated adherent people to placebo or other groups to show dose response for estimator in a few hundred patients at multiple centers, and they cost $10 to $100 million. Extremely difficult trials confirm difficult and very difficult ones, but they also estimate truth about likely effects outside the clinical test frame, the estimand. For that, we need power for mundane subgroups like women and men, old and young, and increasingly for esoteric ones like genetic variants. Excluding parachutes, these kind of studies need about 5 to 25,000 randomized subjects. They cost $100 to $1,000 million or more. It's tempting to believe that big, wide, or deep data, artificial intelligence, and machine learning will come to our rescue. By all means, let's curate data to identify, characterize, and follow subjects. Let's avoid missingness and take out the human guesswork. But when we move from shiny institutions to the mean streets of extremely difficult trials, humanity tends to kick in. Life, and sadly sometimes death, gets in the way. Subjects lose interest or trust. Investigators get distracted. Study teams get overwhelmed. Sponsors get jumpy. Regulators get curious. And lawyers, well, they just get silly. These are emotional, not rational quandaries. Building the medicines company, we performed and planned extremely difficult trials in more than 150,000 people, most in cardiovascular disease. We were ranked #1 for R&D productivity in CNBC's RQ index. We published a couple of dozen papers in the New England Journal, JAMA, and Lancet. We had the good fortune to see all 8 of our phase 3 drug candidates win FDA and EMA approval. Our unfinished opus was a 60,000 subject 5-year primary ASCVD trial framed up with anticipated costs of about $60 million. At the time, Novartis acquired us for almost $10 billion about a year ago. What I learned chasing reliable inference for valid estimates over 25 years is that extremely difficult clinical trials begin and end with purpose. They're not cool calculating scientific instruments, not click, run, read, report, and repeat. They simply can't dehumanize. Instead, they're built for task, multidisciplinary, geographically and culturally mixed enterprises lasting years, with logistics that Jeff Bezos would be proud of, and the human commitment and foibles of hundreds of inconstant professional contributors. I mean, I dare you to count them and their turnover, plus thousands of patients, their families, friends, caregivers, and co-workers, all demanding transportation, venue, and Starbucks, all yearning for emotional connection, recognition, and, dare I say it, tender loving care. In Zen and the Art of Motorcycle Maintenance, Robert Pirsig mapped common ground for Eastern and Western views of quality. what the ancient Greeks called arete, or excellence, and the Eastern philosophers called tao, or the right way. Pirsig painted Plato's preference for reason over emotion as a basis for human progress. Aristotle saw it that way too, of course, but Western thought therefore developed on that basis, so that today we see objectivity, reason, and logic as hallmarks of scientific work, for good reason. But subjectivity, emotion, imagination, intuition, and rhetoric have been relegated to relatively minor roles, especially for extremely difficult clinical trials. But wait, this may be a missed opportunity for, just as Pirsig suggested, when we reconcile scientific, artistic, and spiritual values, we enable quality, which after all is caring. And we engender trust, which is a potent fuel for positive change that, is all too scarce these days. By extolling and pursuing purpose in clinical trials, estimating current and future truth in ways which ensure that those estimates are trusted, we might move closer to the promise of those extremely difficult clinical trials. For those at this marvelous conference, that idea might shake or stir your special leadership, recognizing that control, while essential, is for beginners, and that supporting the dreams of scientists, artists, and spiritualists together can fulfill that authentic purpose. Thanks, Laura.
All right. Thanks, Clive. We'll look forward to more questions in the question and answer. All right, Nevan, you are up.
All right, so thanks, Laura and CERSI. I'm here to give you an update on some of the research that's ongoing in the context of QCRG, the QBI Coronavirus Research Group. This is a group that contains over 40 scientists associated with QBI and UCSF, each with a different diverse background that have come together, I would argue, in an unprecedented way. Ultimately, we're talking about hundreds of different scientists. If you look at all the people in the different labs, and we've broken them down into 12 different subgroups, broken into different biological areas or different technological areas that are all working together to try to come up with solutions for COVID-19. And it's not just collaborations of scientists at QBI and at UCSF in the Bay Area. We've set up a network of collaborators around the world, different academic institutions, as well as different industry connections as well. And I would argue this is one of the silver linings of this pandemic, is to see all these people working together in such a collaborative way, and really to see how fast we can move when we all work together. And just to go through some QCRG papers here, here's one that we had published back at the end of April. We generated a protein-protein interaction map, trying to identify all the human proteins that the virus needs in order to infect our cells, and through drug repurposing, identified drugs that could then be used as antiviral, several of which are in clinical trials, which I'll show you in a minute. We also had another focus on host-directed analysis of infection. We're globally looking at phosphorylation changes, trying to identify kinases that are hijacked in the course of infection, and look at kinase inhibitors as potential antivirals as well. And finally, the last paper, which involves over 200 scientists from 14 different institutions in 6 countries. This is where we used a diverse set of technologies to do a comparison of how different coronaviruses come in and hijack and rewire the host during the course of infection to reveal pan-viral disease mechanisms, with the ultimate goal of identifying pan-viral therapeutic strategies. And just to go into a little bit more detail into this initial paper, as I said, we generated a protein-protein interaction map using affinity tag purification mass spec-based approach to identify over 300 human proteins that were physically connected to one of the SARS-CoV-2 proteins. And through a work led by Brian Shoykat and Kevin Shoykat, they identified 69 different drugs that would inhibit at least one of these human proteins that we think the virus needs to infect our cells. And this led from connections from us and others, out of these 69 drugs and compounds, 26 of these are now being tested in clinical trials for COVID-19. And the one I'm actually most excited about is this one, Aplidin. It's a translational inhibitor that's in phase 3. We're collaborating with the company PharmaMar with this particular trial. This is a drug that had initially been used to treat multiple myeloma. So I'm hoping that you're going to hear about this drug very, very soon. And just a few words here about this comparative pan-coronavirus study. We didn't just look at SARS-CoV-2, we looked at SARS-CoV-1 and MERS. This was the pipeline that we employed for this study. So we generated protein-protein interaction maps with SARS-CoV-1 and MERS to compare to SARS-CoV-2, looking for similarities and differences. We also did a microscopy looking at the localization of each one of these proteins. as we also had antibodies against about half of the SARS-CoV-2 protein, so we could look at localization in infected cells. We did CRISPR-based and RNAi-based functional genetic analysis of all the interacting proteins to determine which genes were genetically required for infection. And we put all this information together and focused our attention on what we thought were key nodes or key interactions for structural characterization. Here's a cryo-EM structure led by Clive Verba and Oren Rosenberg, where we have TOM70, a human protein binding to a SARS-CoV-2 protein, ORF9b. This is a connection we also see with SARS-CoV-1, and we think that this represents a great drug target, and we're looking at this, a pan-coronaviral drug target. And finally, we've been comparing all of our molecular data with previously collected clinical data to see if we could retrospectively see if any of the drugs we're predicting have had an effect on SARS-CoV-2 or COVID-19, and several have. And what I'm describing here is ultimately an infrastructure, not just to study SARS-CoV-2, but the next virus that's going to cause the next pandemic. And I would say that this is an infrastructure that we should be using to study all diseases. And that's my last point here. One of the major problems I have with our scientific enterprise is that it's so siloed. Everybody's focused on just their particular interest. And what we've seen here is during this pandemic is that when you break down these silos across different laboratories, across different institutions, and between academic institutions and pharmaceutical companies, we can move so incredibly fast. And the challenge, I think, for scientists is to keep this spear, keep this infrastructure in place so that we're better ready for the next pandemic, because it's coming. And I would argue we should be studying all diseases in this collaborative fashion. Why aren't we working on breast cancer or head and neck cancer or Parkinson's or Alzheimer's using this infrastructure? So I hope that this spirit stays in place as the dust settles on this pandemic so that we can attack all diseases in a much more fun and productive way.
Well, Nevan, don't worry. I'll be contacting you right after this session, and we're going to get to work on that.
Excellent.
All right. Okay, Jason, go ahead and share your slides.
I'd like to talk today about some unconventional approaches to drug discovery that we've been working on in my lab, sort of almost anticipating the COVID-19 pandemic that we're dealing with right now. So in order to sort of define unconventional, I'd like to talk a little bit about sort of conventional approaches for the development of anti-infectives. So one of the classic case studies that one can use is to think about HIV and development of drugs for treating HIV. A lot of fundamental research was done looking at the viral life cycle, determining what the key steps in the viral life cycle were and what the key proteins were. And the strategy was to basically develop small molecules that could target these proteins underlying these critical phenomena, probably most famously reverse transcription and proteolytic processing, and develop drugs that act on those proteins. And this has been a very efficacious way of treating viral infections. And we can look more broadly at antivirals and see other sort of examples of this, right? So the compounds shown here on this slide were developed specifically to treat, to target proteins that were important for Ebola, influenza, and hepatitis. So this idea of developing specific drugs for specific infections turns out to have its limitations, as we're seeing here in the SARS-CoV-2 COVID-19 scenario. So these conventional strategies of developing drugs for specific infections is just far, far too slow. So one of the things that's happened remarkably is, as Nevan has pointed out, we've learned a lot about the viral life cycle in the past couple of years, but trying to think about developing a drug that targets specific proteins in the viral life cycle turns out to be incredibly complicated and time consuming. And as Nevan indicated, one of the key strategies that people will use is the idea of repurposing. And so probably one of the best known examples of repurposing is taking this drug remdesivir that was developed for the treatment of Ebola, so reverse RNA polymerase inhibitor, and using it for COVID-19. And the results there, it's certainly efficacious, but conditionally so. I don't think it was the game changer that folks hoped that it would be. So this leads me to what I've been thinking about for some time. This idea of having an unconventional approach to treating infections. So can we think about modulating host physiology to prevent a microbial infection? So in this case, we're not thinking about targeting a protein within a pathogen. We're thinking about targeting a protein within the host. So the advantages of this approach are that you can have a small molecule that would have broad spectrum anti-infective activity. And the development of resistance should be slow because, again, you're not targeting something directly within the pathogen. So as we've contemplated this idea, we've thought a lot about the whole process of trafficking in cells. So we know that cells have machinery for moving things in and out, anterograde trafficking, moving things from inside the cell to the outside, and then retrograde trafficking, moving things from the outside to the inside. Turns out that a number of pathogens take advantage of retrograde trafficking for infection. A few examples are sort of shown here of viruses that take advantage of that. So retrograde trafficking, in principle, could be a druggable phenomenon. So if you had a small molecule that could perturb retrograde trafficking, then in fact, you might have a molecule that would have anti-infective properties. So over the years, a number of small molecules have been reported in the literature that intervene with, that perturb this retrograde trafficking. Now, interestingly, most of these molecules were just used as sort of probes of biological systems, but they had not been shown to have any anti-infective activities. So in collaboration with Walter Atwood at Brown University, we were really interested in investigating the antiviral potential of one of these compounds. We started this compound called Retro-2. So in 2014 in InBio, we published a paper describing the antiviral activity of Retro-2 in polyomavirus infections, which was Dr. Atwood's focus. So we were the first to report the antiviral activity of these retrograde trafficking inhibitors. And over the past 7 years, a number of small molecules, a number of cases of prevention of infection by retrograde trafficking inhibitors has been reported. Now, I'll point out here that we have viruses shown here on the top: JC polyomavirus, papillomavirus, even Ebola. Infections are prevented by Retro-2, but it also works for bacterial infections like the bacterium that causes chlamydia, but also against a parasite, Leishmania. So we have really a compound that's apparently has sort of almost a panacea effect in terms of infections. And just last year in May, it was reported that Retro-2, an analog thereof, exhibited superior activity to remdesivir in inhibiting infections of cells by SARS-CoV-2. So this was an important finding. And we thought that we could sort of build on the fact that we had been doing medicinal chemistry around the Retro-2 scaffold for some time and discovered this compound DHQ360, which has remarkable activity against SARS-CoV-2, against polyomavirus in cells. And we've recently worked with a number of collaborators, including Nevan, Roel Andino, and Joe DeReese at UCSF, demonstrating this compound has activity against SARS-CoV-2.
Okay, wrap up.
Now, we've also been interested in Retro-1, and we have reported the discovery of an analog of Retro-1 that has really potent activity against pox viruses. And this compound, again, has activity against SARS-CoV-2 in cells. And we're actually now in the process of doing animal studies with these compounds. So we're really excited about this.
Okay, that's fantastic. All right, so I see that we're getting some questions in the question and answer. While we're starting, I want to ask a question to Clive. You know, in your thinking about developing the PCSK9 inhibitor, which was really trying to come up with a cost-effective, you know, a relatively inexpensive way to reduce cardiovascular events in people who have persistent high cholesterol. You went to the NHS, and and both for your strategy for accruing patients and for getting approval and organizing it, what were you trying to accomplish? And how did that really take years off the development process for this? And did it meet all of your expectations?
We wanted to find patients quickly. That was, and a lot of them. There are 700,000 patients in the United Kingdom who have ASCVD with high cholesterol above 70 milligrams per deciliter, which was the target audience. And they're all actually documented on NHS records, so you can find them. From there, we were able to reach out to primary care physicians directly and recruit them. So yes, it really accelerated finding the patients with specific phenotype and with a willingness to join in a trial with the support of their primary care physician as well as their cardiologist. So that was the first step. The second point was that with the British government, we also went after cheaper manufacturing. RNA is an expensive molecule to make. Typically, we were able to put a consortium together that could bring the cost of oligonucleotide manufacturing down substantially. And then thirdly, quite frankly, we developed exactly what was being talked about in a previous session, which was a pre-commitment to buy the product should the clinical trials work out.
Fantastic.
Which was a 10-year deal for about 300,000 minimum patients a year to be treated. So in 3 dimensions, we worked with the British government to do that.
Because they have a system and you made it cost effective and still profitable.
Well,
So
I mean,
win-win-win. Yeah.
You have to sort of
Yeah.
touch a cap to NICE, who were heavily involved, MHRA were heavily involved, a variety of other British agencies, including their commercial buyers, to create a program that cut across disciplines and was agreed to and contributed to by everybody.
Absolutely remarkable. All right, Dr. Krogan, the question for you, Nevan, was how many of the repurposed drugs or the repurposed drugs that you identified have been tested now in clinical trials, or has that started?
So there's, out of the protein-protein interaction map that I had alluded to, out of the 69 drugs that we identified, 24 are presently in clinical trials. And from our phosphorylation work looking for kinases and kinase inhibitors, there's about 10 or so from that study. So about 35 in total. But the one I have to say, just to reiterate, the one I'm most excited about is this one, Apladine. It's in phase 3 right now. It's from the company Pharmamar, the translational inhibitor, and it's been used to treat multiple myeloma. One of the reasons I'm really excited about it is because of all the drugs that we have looked at here in San Francisco and with our collaborators in New York and in Paris, it's by far the most potent. It's IC90 is a picomolar. It's about 35 times more potent than remdesivir. And in 2 different mice models, we've shown that it's incredibly potent. So I'm hoping that people will hear about this drug very soon.
And is this mostly to prevent severe infection? So at the earlier, you're targeting like allowing the virus to get a foothold?
Yeah, I mean, all antivirals work early. When it's more problematic, it's later. That's when you have inflammation. Although that being said, this drug has been used for multiple myeloma, Apladine with dexamethasone. So it could be a nice combination.
Something maybe we should think about it for iSpy. We'll have you present.
Yes,
okay. That's great. In 2 weeks, we'll have you do it. Okay. Okay, great. Okay, so Jason, I had a question for you. What you described sounds so exciting, but what's the potential downside to targeting retrograde trafficking? You know, what could go wrong?
Well, yeah, lots could go wrong. to be frank, I mean, so the retrograde trafficking is a very important phenomenon in cells. There's no doubt. But one of the peculiar things about the compounds that we have been working with is that they don't appear to completely suppress retrograde trafficking. They appear to basically interfere with the targeting of particular proteins to membranes, and I think it's that selectivity. We don't understand the selectivity of the action of those compounds on those specific proteins, but I think it's that selectivity that gives these compounds some safety. So we have so some of these retrograde trafficking inhibitors have been shown to be efficacious in mouse models of infection. And with some of our more recent work in collaboration with Raul and Dino at UCSF, we found these compounds are actually safe in mice. And so I think there is some promise. So we are in the process of trying to understand how it is that we can just tweak this phenomenon a little bit, but not have catastrophic toxicity to cells.
Be ready for the next pandemic. So maybe Clive, give us a if you think about some of the work that Nevan has described and Jason has described, again, this is trying to prevent these severe infections, the next pandemic that comes along. Should we have a pre-organized agreement for how we're going to kind of launch some of these trials or have it ready to go? Once the pandemic starts, is it too late? Just like it's too late, to do a prevention strategy when someone's critically ill. Oops, you're on mute.
Yeah, the WHO came in for a lot of stick at the beginning of this pandemic that they weren't leading anything, but that's a very unfair thing to ask them to do. It was also apparently unfair to ask the American government to do it on a global basis or any other government to do it. We don't actually have an institution that could set up what we need for Nevan and Jason, and I think that G7 needs to take up that. because it's not going to happen through United Nations. It's not going to happen through NATO, but it is a defense problem. So I think it's going to have to be led at that kind of level. No good doing it just for the US, in my opinion. So I'm hoping G7 is going to come forth with something that everybody can get on board with now that there's a little bit more openness than perhaps there was, pre-election time. So if we can set up that kind of civil authority, then it's possible that these drugs could be developed in the way that Janet and Rob were talking about earlier, in disciplined ways, which are more rapid because there's an established infrastructure. But the idea we do it nationally is not going to work. It'll result in conflicts and confusing data. So job one is to get this global.
Yeah, so Nevan, I think you've proven that you can do that, do this research and discovery in a global way. What do you say? Do you agree that then the next step is to have this kind of global infrastructure, maybe that collaboration sort of then create a clinical arm that's kind of ready to go, that sits on top?
To have, I mean, kind of what Clive's alluding to have another entity come on board and help with funding to kind of set up this would be fantastic. But I would even say, look, NIH or NSF or DARPA, the existing funds could be used much more effectively by bringing scientists together, both on the basic research, discovery research arm, but also connecting the discovery researchers with the clinicians. So I'd like to see NIH put a lot more money into collaborations. They could do a lot better on that front. And the closer scientists are working together, the better we're going to be prepared for the next pandemic or the next disease we all want to tackle.
Yeah, I think so. Jason, if you knew that there was this pipeline ready to go, what would you be doing to sort of accelerate your ability to get ready?
Right. So, when you have chemical matter that you think is interesting, to be able to get it in people's hands and have it evaluated, I think is one of the key things that we'd like to do. And if there's chemical matter that's coming, or that needs to be optimized, that might come out of a screening program to be able to adapt it. So I think that team approach to science would be really quite powerful.
Certainly all the safety testing could be done. It doesn't require a pandemic. All that should be organized and ready. And perhaps, and the same thing, Nevan, even for some of the things you're talking about, just getting the safety pieces of this organized and run, once the vaccine is here and we've sort of organized, I mean, we're just waiting for the next pandemic, but why wait for the next pandemic? These are things that could be tested, all the safety ready to go, so that the minute something shows up, this could be deployed, as Clive said, in this kind of organized infrastructure.
Yeah, and I couldn't agree more. And what I would also argue here is that the reason why we weren't ready, a major reason in my opinion, was because science is really rewarding the individual. The system is set up to often discourage collaboration. One person gets an award, one person gets tenure, one person gets a grant. We really need to change the entire infrastructure and not discourage collaboration, but encourage. And that's particularly pertinent, I would argue, for young scientists. They have the best ideas. We got to bring them into the fold and reward them for sharing. And until we change this, we're probably going to be back to square one for the next pandemic. So I hope we've learned a lot through this, and not just how to work together, but how to set up infrastructure that would facilitate us working together.
Well, I think that's true for both team science, but I think, it's also interesting for us to think about, in the United States in particular, we've had this huge, argument about infrastructure and whether we need it. And I think you compare sort of what Clive did in the NHS, or the ability for the recovery trial to get up. They have 2,000 patients a week on. We've, in record time, put up I-SPY. We now have 50 patients a week, which is actually remarkable for the US, but not remarkable compared to what we could be doing. And, because every hospital is the same. I mean, at every level, we have to think about organizing for value. There's so much waste in this system.
And related to that, did we really need 400 clinical trials on hydroxychloroquine? Was that really a good use of funding? I mean, that was
or the same number on therapeutic plasma without one control.
Exactly.
I thought Janet's data this morning were quite revealing, and I think perhaps of all of us, she's had the best view of what's going on. 2,610 trials, I think she mentioned, that they've put into this data lake at the FDA. Only 5% met normal criteria of power and randomization. That's such an incredible waste of time by the other 95%.
It's
I
very
think
sad.
it is driven by the current motivational strategy for academic tenure.
Yeah.
I think it's appalling, and I think the industry and academia should really take it as a big lesson. But more importantly, or just as importantly, I should say, I think governments have to recognize that only they really have the integrative power to force major change in this. If we try to do it bottom up through academic routes, it's going to be a bit slow.
I agree. And again, that's as we talked about on my panel, is if you organized a different way of doing clinical medicine. I mean, we're practicing the way we did in the 70s. My goodness, if we had a system of mission critical data that we're collecting as the point of care, imagine how much easier it would be to do all of this, So anyway, I know that this has been a fantastic discussion. We have now about 8 minutes break. And we're going to start the session back up. It's supposed to start at 12:20. Hopefully they'll give us an extra 5-minute break. I want to thank Clive and Nevan and Jason for fascinating contributions and for this really fun discussion. Thanks so much, everybody.